Hi everyone, today we are presenting on the JACS article of Structural Basis of Sirtuin 6-Catalyzed Nucleosome Deacetylation presented by Elia, Gionil, Nimika, Caroline, and Nicole. Reversible lysine acetylation modifications represent a vital class of post-translational changes that exert a profound regulatory effects on chromatin structures, gene expression, and various biological processes. This intricate relationship between acetylation and deacetylation is guided by a group of enzymes known as histone deacetylases, HDAC, which includes both metahydrolase HDAC and NAD-dependent HDAC, called strutins. Dysregulation of these HDAC has been implicated in diseases like cancer, prompting extensive research in the pursuit of HDAC modulators. This paper explores how strutin 6, which is shortened to SKIRT6, a long enzyme containing 355 amino acids, plays a role in DNA damage response and modulating post-transcriptional pauses, releases, and altered activity in various diseases. This paper will test how SKIRT6 can deacetylate histones more efficiently in chromatin substrates. This study aims to address these gaps by assessing the deacetylation rates in specific lysine sites in histone H3 and histone H2B within site-specifically acetylated nucleosomes. Furthermore, the use of protein semisynthesis has enabled the preparation of specialized nucleosomes, such as lysine 9 thiocarbonyl-derived nucleosomes, to trap SKIRT6 nucleosome complexes.
Acetyltransferases and deacetylatases control the acetylation of histone-lysine residues. The acetylation of histone-lysine residues is a post-translation modification that is important for chromatin structure and gene expression. Humans have 18 deacetylases, 7 of which are sirtuins, which are enzymes that are NAD-dependent. SIRT6 is a sirtuin enzyme that helps with genome stability and gene expression. It is found on histone 3 and is NAD+ dependent. It has been found that with an increase in SIRT6 activity, the lifespan of fruit flies in mice seems to increase. SIRT6 is known to catalyze 3 different reaction types: lysine deacetylation, long-chain lysine deacetylation, and ADP ribosylation.
SIRT6 has functions in other catalytic activities as well. For example, deacetylation of long-chain molecules and mono-ADP ribosylation of proteins. SIRT6 has been found to be important in maintaining homeostasis in cells. It plays a large role in metabolic processes and DNA repair. Dysregulation of sirtuins can cause diseases, which is why this study aims to identify HDAC modulators. The hypothesis of this study is that SIRT6 is better able to deacetylatate histones in chromatin substrates.
I will now explain the different methods that were used and the steps that were taken. There were 4 steps total in this process. The first was the cloning, expression, and purification of the SIRT6. The second step was general deacetylation. The third method used was cryo-EM structural analysis of the SIRT6 nucleosome complex. And lastly, they looked at the SIRT6 mutants activity and EMSA, which stands for electrophoretic mobility shift assay. This was done at a Q5 site-directed mutagenesis at New England Biolabs, and overall what they did was they analyzed site-specifically acetylated nucleosomes as substrates to assess the rate of deacetylation of a range of list sites in histone H3 and histone H2B. They used a protein semisynthesis to prepare a list 9 thiocarbonyl derivatized nucleosome to trap a SIRT6 nucleosome complex. First was the cloning, expression, and purification of the SIRT6. What they did was they grew the cells at 37 degrees Celsius and 200 rotations per minute to an OD600 of 0.6. Isopropyl was then added and the cell grew to 0.5 millimolar, and the overexpression was induced. This grew them to an additional 18H at 25 degrees Celsius and 200 rotations per minute. They were then put in an ice-cold lysis buffer, which was made up of 20 millimolar tri, 500 millimolar of sodium chloride, and 20 millimolar of imidazole, which had a total pH of 7.5. It then was lysed with a French press, which is where the outer boundary is broken to release inner cell materials like DNA, RNA, and proteins. The lysat, which is the inner cell materials, was then clarified by centrifugation. Centrifugation separates molecules having different densities by spinning them. It was then applied to a pre-equilibrated resin and then incubated. The resin was drained and was washed with 10 column volumes of a wash buffer, 25 column volumes of high salt wash buffer, and then eluted with 10 column volumes of an elution buffer. The unclean protein was removed by a second passage over resin. The flow-through was diluted with 10 milliliters of heparin buffer A and concentrated by a spin concentration. It was then applied to a heparin column and eluted with a linear gradient from 0 to 50% heparin buffer B over 20 column volumes at a flow rate of 0.6 milliliters per minute. These fractions were checked by Kumasi staining following SDS page, and the pure fractions were collected. This served as the quality check for WT SIRT6 and all of the SIRT6 mutants. It was then further purified using a Superdex 200 column with a Superdex running buffer. The pure fractions were concentrated to 40 micrometers. The WT CERT-6 was characterized by an intact electrospray ionization mass spectrometer and deconvoluted using UNIDEC. All CERT-6 constructs were separated and flash frozen in liquid nitrogen and then stored at 80 degrees Celsius prior to further use. The next process was general deacetylation. The free histone proteins were diluted in the CERT-6 reaction buffer and kept on ice. To start the reaction, the CERT-6 was added. A small amount was removed every 30 minutes, and each quenched sample was then boiled and then resolved on an SDS page gel. The gel was transferred to nitrocellulose membranes for 5 minutes and then blacked for 1H, then incubated overnight with a primary antibody. The site-specific antibodies for H3, acytylated H3, H2B, and acytylated H2B were listed. The HRP-linked secondary antibody was applied for 1H at room temperature. Membranes were washed and treated with an ECL substrate reagent and then visualized with a mini gel imager. The band's intensity was able to be quantified. The intensity values were divided by the intensity value at time 0 to get the relative intensity, and then fit to a single-phase exponential decay curve, and the kinetic parameter was calculated. Next, cryo-EM structural analysis of the CERT-6 nucleosome complex was done. The CERT-6 and NAD were mixed and then purified by size exclusion chromatography. This purified complex was mildly cross-linked with glutaraldehyde and dialyzed. A 15,192 micrograph dataset and a second dataset with 6,922 micrographs and a 30-degree tilt were both collected on a Titan Krios. The first dataset picked 2,507,720 particles, and the second dataset picked 1,556,859 particles. After 3D classification, 95,205 particles were refined to a final resolution of 3.1, which produced map A. Further classification allowed for a second map to be made at 3.3 resolution. Finally, they built a model by rigid body fitting in the nucleosome, CERT-6, CERT-6 residues 1 through 16, and the H3 tail. The last step was looking at the CERT-6 mutant's activity and EMSA. The nucleosome binding of CERT-6 constructs was assessed by electrophoretic mobility shift assay. CERT-6 constructs were diluted to a working concentration of 9.6 micrometers, and then serially diluted to 4800, 2400, 1200, 600, 300, 150, 75, and 0. H3K9AC nucleosome 185 BP was diluted in the CERT-6 binding buffer, and then combined with the CERT-6 enzyme to a final nucleosome concentration of 20. Then, they added NAD to a concentration of 150 micrometers. Different samples were prepared to get a final CERT-6 concentration of 960, 480, 240, 120, 60, 30, 15, and 0. Samples were incubated for 30 minutes on ice, followed by native electrophoresis on 4 to 20% TBE gel run at 130 volts for 110 minutes on ice.
This is an explanation of Figure 1. In the first section of Figure 1, they showed the histone deacetylation sites in the semi-synthetic histones that were made, that is the H3 and the H2B, which were then assembled into their respective nucleosomes at base pairs 147 or 185. In this figure, they compared the acetylation at these two sites as well as deacetylation of free histones. As we all know, a nucleosome is composed of DNA coiled around the histone. Figure 1b shows the Western blot analysis for the deacetylation of the H3K9AC protein, so the free histone. And this figure shows that there was no deacetylation of the free histone. Figure 1c shows the Western blot analysis for the deacetylation of the H3KAC, the H3KAC nucleosome deacetylation at the various acetylated points. Figure 1d shows the Western blot analysis for the deacetylation of H2B KAC nucleosome at position 147 base pairs. As we can see from the figure, there was no deacetylation at either of the acetylated sites, whereas in the analysis or the Western blot analysis of the H3K acetylated points on the 147 base pair nucleosome, we do see deacetylation occurring, that is in Figure 1c. Figure 1e shows the comparison the comparison, that is a plot of the comparison of the rate of deacetylation over the enzyme concentration for the different H3 KAC and H2B KAC acetylated points. As we can see from the plot, only the H3 KAC acetylation points showed different rates of deacetylation. The H2B KAC acetylated points showed no sign of deacetylation. Figure 1F shows us the same data for the velocity of or the rate of deacetylation compared to the enzyme concentration of the free histone protein. Figure 1G shows us a curve fitting for the rate of deacetylation for the different H3K9 AC sites of the nucleosomes. And as we can see that there was an indication of deacetylation at positions K27, K23, K18, K9, and a little bit at K14. Whereas in figure 1H, where it shows the curve fitting for the rate of deacetylation for the H3K9 free histone, we can see that there was no significant deacetylation going on in that figure. This figure also shows us in one of the supplementary figures that there was also deacetylation of the H3K9AC nucleosome at positions 185 as well. And so both the H3K9AC nucleosomes were deacetylated, whereas the free histones were not acetylated and were not deacetylated. And then the H2B-KAC sites were also not deacetylated for both the free histone and the nucleosome. Figure 2A and B show the rate of deacetylation, that is the Michaelis constant plot, as a function of substrate concentration for the nucleosome at 147 base pairs and the nucleosome at 185 base pairs. Deacetylation, obviously this showed deacetylation, and the Michaelis constant was between 17 to 19 nanomolar. This figure also shows that deacetylation was not dependent on the length of the nucleosome. Previous literature shows that the rate of deacetylation is dependent on the concentration of NAD plus, and that different saturanes show different NAD binding nodes, and this binding is dependent on the specific acetylated lysine substrate. So they tried to compare the rate of deacetylation as a function of the concentration of NAD plus, and they got a Michaelis constant for that plot at 150 micromolar. There is a part of the figure that wasn't shown in this figure, but was shown in the supplementary, where they checked the effect of ionic strength on the binding of SAT6 or deacetylation of SAT6. It shows that the introduction of sodium chloride decreases the rate of deacetylation. They also checked the effect of an activator molecule, methyl 2N5 bromo 4 fluoro 2 methylphenyl sulfonoyl 5,3,5 dichlorophenyl sulfonamido benzoate, that is the MDL 800. The figure for the molecule, or the chemical structure for the molecule, is shown in figure 2E. Previous literature reports that this molecule allosterically activates SAT6, and they try to test that using a Western blot analysis to check the effect of the molecule on the rate of deacetylation for H3K9AC 147 base pair nucleosome substrate. And they did this with the addition of DMSO. They did notice that DMSO reduced the rate of acetylation, and that this molecule as a whole improved the deacetylation, the SAT6 deacetylation process. Figure 1F shows the rate of deacetylation at varying concentrations of the MDL 800 as a function of time. We do see an increase in the rate of deacetylation with an increase as you increase the concentration of MDL 800, and as well as we also see that increase with time. Figure 1G shows the curve fitting for the activation of SAT6 by MDL800. And it said the study says that the EC50 was reported at 3.5 micromolar. EC50 is the concentration of MDL800, which activates or improves the deacetylation of SAT6. And as we can see from the plot, there is an increase in the activation plot for SAT6. We are trying to understand the selective binding of SAT6 to the H3K9 AC nucleosome substrates. They prepped modified H3K9 AC with some thiocarbonyl groups, that is thioacetyl, which they designated as SAC, and thiourea, which they designated as MTU. These two installations from previous literature were shown to be inhibitors of SAT-twins. This is because they can trap the enzyme and the substrate complexes due to the slow turnover of ADP glycoside, that is versions of ADP ribose intermediates. So the figure 3a and figure 3b just shows the pathway for this the chemical reaction for this turnover. It's important to note that this process of slow turnover, where you trap the enzyme and the substrate complex, can be used in determination of structural determination of SAT-twins bound to their substrates. So for figure 3c, they did a Western blot analysis to show that even with the introduction of SAC, SAT6 still deacetylated H3K9AC at 185 base pair nucleosome. So the introduction of SAC did not affect the acetylation of the nucleosome. Figure 3D, they tried to compare the deacetylation rates of the H3K9AC and the modified H3K9AC, that is the H3K9AC that had SAC introduced into it. There was no significant difference in the acetylation rates. For figure 3E, they modified the H3K9, the modified H3K9 ACMTU mutant resisted deacetylation. This was shown using an electrophoretic mobility shift assay, and they did also show that this resistance was dependent on the presence of NAD+. For figure 3F and 3G, they show the ESI mass spectrometry plots for the trapped, which shows the trapped analog of one of the proposed deacetylation intermediates. That is the N13 oxathiolan 2 ylidine amine ADP ribose adduct.
The vehicle appeared the chiral structure of SAT6 nucleosome complex. We can get the single particle chiral data by using the Titan Crios microscope with a Casri direct electron detector and energy filter. This structure consists of 95,000 particles with an overall resolution of 3.1 angstrom. We can understand this size by comparing to the size of nucleosome or SAT6. In the chiral EM data, we can observe the three parts. The first thing is the SAT6, which is located on the H2A C terminus. In the SAT6, there was a small change compared to previous crystal structure. However, we can reserve the N terminus, and the thing we can reserve is only a small part of the SAT6 terminus. The second thing we can observe is the ADP ribose intermediate analog moiety in the SAT6 Rosmaford. The last thing is the histone tail between the nucleosomal DNA and the SAT6. SAT6 interacts with histone and nucleosomal DNA. First, SAT6 interacts with the exit patch of nucleosome using zinc binding. The zinc binding is unique property even between other SATins. The loop bind by zinc binding is observed. For example, K170 reaches to E64 and R172 touches the E56. At the last, the R175 connects with E61, D90, and E92. We can test this interaction by using mutation. We use three kinds of mutation. The first thing is R172A mutant, and the second thing is R175A mutant. The last thing is mutant with K170A, R172A, R175A, and all 178A. By watching the change of deacetylase activity and ability to bind the nucleosome depends on this mutation, we can understand how this zinc binding interaction is working. In the figure 6, we observe 15 base pairs of nucleosomal DNA from SHA5.5 to SHA7. These base pairs are unwarted from the histone octamer, and SIRT6 interacts with this unwarted DNA. For example, we can observe the 4 base acids. 3 base acids interacts at SHA6.5, however the all 248 interacts at SHA7, and all these 4 things are interacting with the phosphate backbone of the unwarted DNA. Also, we can check the mutation of these. The mutation of all 231 and 32 results reduction of deacetylase activity and a reduced affinity. However, because of the SIRT6's position, the mutation of all 248 doesn't show a big change. Also, we can observe the interaction formed by SIRT6 at the C terminus of H2A. Observe the ADP ribose intermediate analog. The adenosine moiety of cofactor is interacted at A site, and the reactive ribose moiety is interacted at B site. The A site and B site can be observed at figure 5A. Compared to previous structure, the SIRT6 has larger lesion acetylation and can complete deacetylation. Also, we can find the H3 lucidK4 insert itself into the groove of DNA at SHL6.5. This interaction with DNA has stabilized the H3 tail. At the last, we can predict the H3 tail residue K14's interaction because of this distance, because the distance is enough to reach the active site of SIRT6.
Facilitation of histone substrates by SIRT6 is very selective and efficient. SIRT6 has a preference for histone H3 nucleosomes over the substrates, including H2B. This is because H3 is in a favorable position for deacetylation. H3K9AC, H3K18AC, and H3K27AC are most efficiently deacetylated sites by SIRT6. SIRT6 has very important interactions with the nucleosome. It binds to the acidic patch of the nucleosome, stabilizes unwrapped DNA, and interacts with H2A residues.
After discussing as a group, we found that the article was hard to follow, disorganized, and that the data was difficult to understand. We suggested that using the H3K27AC, which is a SCRT6 deacetylation site, should be considered for future studies. Some discussion questions to consider